Hi everyone, I'm Doody from University of South Florida, and I'm going to talk about synchronous hatching and emergence in turtles, and I'll especially refer to some adaptive hypotheses for why it may evolve in turtles. And if I can get this to page down, we'll keep going. Fun fact, so did you know most of what we know about hatching and emergence in reptiles is from turtles? So, and this is because turtles, some of them are gracious enough to leave a track in the sand, like this spiny soft shell mother. Of course, some turtles are selfish and nest in vegetation, making it harder to find the nest. But the bulk of what we know about hatching and emergence in reptiles is from turtles. We don't know much about lizards in this respect. When was the last time you went out and looked for snake eggs? Never, right? Crocodilians, they're conspicuous nesters, but they always lay in places that are remote and hard to get to. You got to take airboats and stuff to get there. Then you have to deal with the mother, right, defending the nest. So we owe turtles for what we know about hatching and emergence. So what is synchronous hatching and emergence? This is going to evolve the definition as we go through the talk. Hatching and emergence are separate behaviors, right? But they can be linked, they tend to be linked. So you could emerge synchronously after hatching asynchronously and vice versa in all the different combinations. But traditionally, collectively, we talk about them as a simultaneous escape of offspring from the nest or nest sites. This can occur within or among nests, but I'm going to focus mainly on within nest hatching and emergence today, and you'll see why. And traditionally, we've considered these behaviors important anti-predator behaviors that are related to fitness. For example, the classic example is the mass emergence, hatching emergence synchronously of sea turtles, right? So each one of those sea turtles has its predation risk diluted, safety in numbers because it's surrounded by so many others in the face of predators such as crabs, raccoons, cats, dogs, armadillos, foxes, birds, and then of course, as all these turtles get to the ocean, they face a gauntlet of another suite of predators, fish, dolphins, etc. And there is evidence that this predator dilution, this predator swamping actually occurs, experimental evidence for it. But what about single nests of turtles other than sea turtles? So the picture you're seeing is all about among nests, synchronous hatching and emergence in sea turtles. What about a single nest of a non-sea turtle? Is there synchronous hatching and emergence? I'm going to use Pig-nosed turtle story as an example because it's what got me into this subject area. It's extremely interesting and unique, and I think we would expect, based on this story, to see synchronous hatching and emergence in this species. So pig-nosed turtles in northern Australia lay eggs during the dry season. The eggs complete development in the dry season, but they do not hatch in the dry season. They go into this suspended animation where they're waiting in the egg for the onset of the next wet season. And so when we get heavy rains and rising river levels over the nests, the eggs then hatch. They've had to evolve explosive hatching, and you can see this on Life in Cold Blood from BBC, whereby pig-nosed turtles can hatch in minutes instead of hours and days, which we see in most turtles. So we've known for a long time that hypoxia is a cue for hatching in pig-nosed turtles. So the eggs go underwater, they become hypoxic, the embryos start moving around and hatch within minutes. In the late 90s, during my PhD, we discovered when we were digging up late-term eggs, that they were hatching in our hands. They were hatching in our hands, in the boat, in the car. It was a mess. And so we had discovered inadvertently a vibration cue for hatching. And we wondered why that would be the case. And as I held an egg and could feel an embryo moving inside it, I thought maybe this movement can expedite hatching in pig-nosed turtles. And so we did some experiments, and indeed we were able to show that groups of pig-nosed turtle eggs hatch and emerge faster in experiments than solitary eggs. And if you plug those data into a model using different river level rises, rates of rises, you'll find the bottom right graph that turtles that are exposed to a vibration cue will always hatch sooner than those with only a hypoxia cue. So basically what we're saying is that the water level starts to cover the bottom of the nest, causing hypoxia, and those embryos moving around instantaneously send a vibration cue through the nest so that they can all hatch faster and get out in a flooding situation. So why did I drag you through an example that was published 7 years ago? Well, I think it's fair to say that we would expect synchronous hatching and emergence in this species given that scenario. And yet when we put cameras out on the nests, that's not what we found. So here are a couple of nests being monitored by the old Trailmaster camera systems. And you can see one pig-nosed turtle hatchling emerging in each picture. On the left, after heavy rains. On the right, after flooding of the nest. In fact, we lost those boxes from that flood, but of course the camera is separate here, so we were good. The interval between emerging hatchlings for pig-nosed was 12 minutes. So we didn't really see synchronous hatching and emergence or synchronous emergence. Some hatchlings did follow 1 minute after another. Some took days to get out. So that was a bit surprising, and that I think kind of sets the theme for the rest of this talk, which is going to be reviewing the evidence for synchronous emergence in turtles in nature, and then using that evidence to scrutinize the adaptive hypotheses for why synchronous hatching and emergence evolves or is maintained. So what are those hypotheses? The first one we already talked about, predator dilution. Goes by a bunch of different names, gets very technical: attack abatement, predator swamping, predator satiation. Although it has a bunch of different mechanisms, it's essentially safety in numbers. A second hypothesis called social facilitation came out in the 60s to help explain how sea turtles, hatchlings get together and in a collective fashion to excavate themselves and escape the nest. And some work out of David Booth's lab built on that recently and showed that there's a huge savings in energy in terms of the residual yolk in hatchlings due to that facilitation. So that's a really important hypothesis, at least for nests that are deep with lots of eggs like sea turtles. The next hypothesis is so little visited that it didn't have a name. So I've given it the name predator preclusion. And here we're talking about the fact that if you have, let's say, a painted turtle nest and your first egg hatches, it's going to give off hatching smells or odors that could attract olfactory-driven predators to the nest. And so it'd be best if everybody hatched and got out as soon as possible, i.e. synchronous hatching and emergence, so as not to attract predators through an asynchronous hatching scenario. And we'll return to that later. Finally, some of you might have already thought, do we have to have an adaptive hypothesis here? If we've got a turtle laying eggs that are experiencing similar temperatures, similar incubation periods, hatching about the same time, why do we need some adaptive explanation? And in fact, that is a good point for small turtles with small eggs and small clutches. In those turtles, we have very little layering of eggs. And so they will experience the same temperatures, developmental rates, incubation periods, and we would expect them to hatch and emerge at about the same time. And so things like box turtles, mud turtles, sabine map turtles, all the small turtles that lay very few eggs or small eggs, we might expect synchronous hatching and emergence without any adaptive explanation. And if that's all we had with small turtles, then the talk would be over. But of course, we have lots of larger turtles with larger numbers of larger eggs. And so when we get these species, the three-dimensional space occupied by the nest is large enough to experience thermal gradients. That is, top eggs are going to be warmer than the bottom eggs. So mean temperatures are going to change moderately, maybe just a bit, but the fluctuations, the daily fluctuations in temperatures are going to change quite dramatically between the top and the bottom of the nest. And so we're going to get top eggs potentially hatching sooner than bottom eggs. And yet in the lab, especially, researchers have discovered that there is still somewhat synchronous hatching. So we've got these thermal gradients. We should have these big differences in incubation period and timing of hatching. And yet a bunch of studies have shown that in fact, that difference is not very large. So Spencer et al. Were the first to discover this with Macquarie River turtles. If you take eggs and incubate them at different temperatures and get some at going faster at a hotter temperature, and then you join the eggs back together, those least advanced siblings will catch up a bit with the warmer eggs that are ahead of them, up to 2 to 5 days earlier than controls. They still lag behind the others by 4 to 6 days, so it's not perfect, but there is some catch up in some species. And then a more recent study showed, instead of catch up, that some painted turtles can hatch at a less advanced developmental stage than other eggs. So there are different mechanisms here for trying, it seems, to synchronize hatching and potentially emergence. It's not really where I want to go with this review, and there's another interesting layer about how they might do this. They can possibly detect one another's heartbeats. Of course, we now know there are vocalizations, at least in Podocnemus expansa embryos, so there's a lot of neat stuff there. I just wanted you to understand the sort of framework behind why we might be extra interested in synchronized hatching and emergence. So what I did was looked at the data for 24 studies on 16 species of turtles. This is emergence data from within nests. The quality of the data are highly variable. Sometimes it's someone checking a nest once a day or once every other day. Sometimes we have cameras getting timestamps for every time a hatchling emerges. I ended up with 3 types of data or 3 sort of parameters. The percentage of nests in which all of the siblings emerged within 24 hours. The second one is just the range of that. So over how many hours or days did all of the siblings hatch and emerge from a nest. And then finally, for a couple of studies, with cameras, we had mean sibling emergence interval data. So let's look and see what we got. For the mean proportion of nests in which siblings emerged, all siblings emerged in 24 hours, we got 47%, so just under half of nests hatched with all siblings within 24 hours. And that's from 18 studies. The range is about 1 to 77 days for all studies, but the mean, if we look across studies, the mean of that is 1 to 17 days. So on average, it took between 1 and 17 days to get everybody out of the nest. And then finally, for the two studies I mentioned, we had 12 minutes and 218 minutes as intervals between, average intervals between emergent hatchlings. And so from this, we can see emergence from these 16 species was not particularly synchronous when we define synchronous as within 24 hours. And certainly 2 or more siblings would not be simultaneously visible or detectable by a predator. Okay, so that says something about predator dilution. There were no clear patterns, even though the sample size is not huge. There were no clear patterns between, say, terrestrial, freshwater, sea turtles, but there was a bit of a pattern with the proportion of emerging hatchlings within 24 hours was high in pig-nosed turtles and soft-shell turtles, and that may just be related to a nice sandy nest that's easy to nest in, easy to emerge from. So what do we say then about our hypotheses, our adaptive hypotheses for why we might see synchronous hatching and emergence? Well, from these data is emergence in turtles, other than sea turtles, is not generally synchronous enough to dilute predation risk. So we can kind of X this out as a generality. Maybe things like Podocnemus expansa with large nests and synchronous emergence could dilute predators. The social facilitation hypothesis seems unlikely to me that this would work for your standard turtle nest, shallow nest with a small clutch size. Again, it could be that things like Podocnemus do use this, and the data in this study were not designed to test that hypothesis. So I've put an X with an asterisk there. The third one I think holds promise. I think we certainly can't, with these data, reject the predator preclusion hypothesis. Remember, that's the one where hatching smells are released with the first hatching, and if everybody gets out, it won't attract, it'll attract less predators to the nest. So this one, I think there's a lot of studies out there now looking at what cues raccoons, animals like raccoons and foxes are using to find turtle nests. I think we can add this to that and look at whether or not the synchronicity of hatching and emergence in turtles matches the sort of abilities of predators to detect and destroy turtle nests. So I think that's the fertile area for research in this area. And so I've given that a question mark. And then finally, it's highly likely that very small turtles with small eggs, small clutch sizes, that there will be a degree of synchronicity in hatching and emergence without invoking an adaptive hypothesis. Now, some critics might say, well, Sean, we never thought that there was synchronous emergence in other turtles besides sea turtles. You're raising a straw man. But there's a number of, a spate of studies that have not only mentioned that synchronous hatching and emergence is common, widespread, and even ancestral, but they've used predator swamping as the context to set their studies into. So it's not me that's raised any straw man. If there is a straw man, it's the collective of these studies that have raised it. But one of the things that comes up as a problem, issue, if you will, is the fact that we lack a good definition of synchronous. So I think we need a new definition of synchronous. So it's often not carefully defined in most of those studies, even the ones that address predator dilution. Some researchers, including myself, have arbitrarily defined it as 24 hours. Whether or not that's particularly useful, I don't know. Certainly not with predator dilution, that's too conservative. Spencer and Jansen have created a definition for synchronous hatching that is understandable given what they were chasing in the laboratory. But I think it's getting so far from what synchronous really means that we need a new definition and perhaps some new terminology going forward. The other thing we need going forward are getting game cameras on turtle nests. I would have thought that this would be happening at a higher rate than it is. Then we can get accurate emergence data. The cameras are down to $50, $100, so this is really doable stuff. So hopefully this data set will grow in the near future. And then again, I think the predator preclusion hypothesis is the fertile area for understanding synchronous hatching emergence in the field and the fit of adaptive hypotheses to that, to those parameters. Okay, that's it for me. Happy to take any questions. Quick plug. There is a chapter in our book that's coming out next year on social lives of reptiles. There's a chapter on synchronous hatching emergence in reptiles. So if you find this area interesting, that'll be perhaps a reason to get the book. You can pre-order it now on Amazon. So I'll try to wait here and see if we get any questions.
There should be some on the way. Just watch your chat, Sean.
Yeah, I'm having a little trouble getting there it is. All right, stack of questions. Here we go. Question 1, apologies if you've answered this already. What did you do to stimulate the vibration cue? Well, initially we discovered it was handling, and then we did some lab experiments where we put them on lab shakers. Pretty crude, but it sort of did the trick. And there's a whole sort of, there's a lot of work in this with amphibians, and my lab's doing it with reptiles, trying to find the right way to vibrate an egg, in many cases to simulate predation, but you know, it's tough to find the right cue when you don't have an actual predator. With the amphibians, with the frogs, Karen Workington's lab uses actual predators, which is the best thing. Question 2, Ricky Spencer. Smaller turtles lay closer to the surface, so any layering will have significant temperature differentials, thermal gradients. It's a gray area. So, you know, the review I did was with all reptiles, and so dealing with small skinks, for example, there's not any significant differences between two small skink eggs that are sitting next to one another. And there's a continuum from that going right up to a sea turtle nest. So when you say significant differential thermal gradients, we don't know where along that continuum it suddenly becomes significant, and that we would expect something like a catch-up that you showed in the lab with a moderate-sized turtle. How am I doing for time, guys?
You're great. Take your time.
Okay. Question 3, how might animals that live in satellite nests fall into the context of this? Would the main chamber perhaps synchronize differently from other chambers? Haven't thought about the triple nesters. The satellite nests tend to be shallower. So yeah, it's a tough one, but they only tend to put one egg usually in the side chambers. So not sure how to answer that one. Question 4, Ricky again. Experimental protocols different in many of the studies, some touching, some not. They may take cues throughout development to hatch synchronously and emerge quicker. Yep, so wasn't really in the scope of my review to get into that stuff, and it's fascinating. So I'm just focusing on the emergence from the field. But good point on the mechanisms, and I hope you guys crack it. Another question from Ricky. What about internest communication and synchronous development? Yeah, a possibility. I think for most turtles, it's not an issue, but could be for certainly species that lay communally. Okay, I think that's it. Thank you for your time, and thanks to TSA.